So there's our nucleotides. Great job, everybody. Adenine, guanine, cytosine, and thymine. We know apple tree, put your car in the garage. U2, like the band, U2. And that's, we know uracil replaces thymine in our RNA. So the way that our blueprints work is simply, how are these nucleotides arranged? Do we have repeaters? And in fact, it's the repeaters that give us our dominant traits. I find genetics kind of interesting, which is genetics really is just the study of the structure of your DNA and how was it assembled, and how can we alter it in future generations. So it is the repeaters that give us what we call dominant traits. And what a dominant trait is something that is outwardly visible. My eyes are brown, therefore the dominant trait in my DNA is brown. But we also have what are called recessive. And these are kind of little backroom blueprints. My body carries the instructions for them, but I don't exhibit. So I have brown eyes, but I may carry the gene for blue eyes because my dad has blue eyes, my mom has brown eyes, so I carry the blueprints for both, which means if I had an offspring, it would be a 1 in 4 chance of them getting brown eyes or blue eyes, because I carry the instructions for both. That's kind of, I think yesterday we were talking a little bit about the slippery properties of canine DNA. And that's how we can take the traits from other breeds and breed them in and take a recessive trait and make it dominant. And that's how, I think yesterday, did we talk about the Newfoundland? And how
they changed the color.
We changed the color and we gave them a little bit more of a white coat. But the one thing we did not want to do is take away like their ability to swim. We didn't want to take away any of the density of the coat. So what did we do? We mixed a little bit of a breed that had the same basic dominant traits, just had a different color. And now we have a black and white Newfoundland that we can now see in the very dark water.
Do they have the double blue claws just like the Great Pyrenees?
So they, plus or minus. Some do, and we'll usually remove them on the Newfoundland, not on the Great Pyrenees. Don't remove them on the Great Pyrenees.
DNA as well, like the dew claw, or does that just?
So yeah, that's DNA, and okay, but here's the problem, and then I'll get off of this and we'll finish this up. Every time you manipulate the DNA, okay, I'm like, I have this dog. Let me take golden retrievers as an example, because you guys are in your dog breeds now, and this is really fascinating. I've got a golden retriever. It is literally the perfect family dog, right? They're amazing, well, until they hit the age of 2. Before 2, they're kind of stupid. But no, they are. They're kind of stupid, kind of destructive. They don't know their own size. They don't know their own strength. After, like, the minute that they hit 2 years old, they just like, now I'm the perfect dog. Beautiful golden color, hence the name. And so what did we do? Well, humans. We look at this perfect golden gorgeous dog and go, wonder what it would look like if it was red. And so we started breeding in the red coat color from some of our terriers, like Irish, didn't know the word Irish, like our Irish terriers. The problem is that every time you breed in a dominant, what are we also breeding in?
Their personality.
We're breeding in, I call them evil spirits. Evil spirits that you didn't really intend on making part of this animal. The red color in our animals who are supposed to be red comes with it the trait for anxiety. Redhead, now I'm talking dogs, okay? I'm not talking, well, technically people too, kind of. Redheads tend to be very anxious. Your Irish setters, your Irish terriers, any of your redheaded dogs tend to be a little bit high strung. That gets amplified in the golden. So the red goldens, we've bred into them a form of schizophrenia.
That's what she told us yesterday.
Yeah, they were kind of
Yeah.
bipolar, the real.
The red ones, oh, there is nothing in my mind, nothing more dangerous than a red golden.
Yes.
Nothing.
I have a red golden doodle and he's so sweet.
Okay, but that's a golden dude. That's a mixed breed.
But his mom is a red golden.
Okay, and
That's what he got the red color.
right. Now, am I saying that they're over in the corner, frothing at the mouth, back crap crazy? Of course not. But what I am saying is they will turn on a dime. I've had 3 dogs in my entire career, and I've had a pretty long career, longer than some of you have been alive, I'm sad to say. I've had 3 dogs in my life that I was afraid of. One of them was a Rottweiler Boxer mix. One of them was a Springer Spaniel, and one of them was a Red Golden.
Yeah, she said Springer has Springer Rage.
Springer Rage, and that's why I was afraid of it, because this dog did have Springer Rage. And the Red Golden is very similar to that Springer Rage, where literally the dog is like, I want you to pet me, I want to kill you. And they will turn just like that. I know, it's terrifying. I had
tough love.
the worst experience with a Red Golden. I had the dog, and it was gorgeous, humongous dog. And he was just like, I want you to pet me. I'm talking to the owner, and the dog's kind of pawing at my leg, and you know how they do.
Trying to get her attention.
Trying to get my attention. So I turned to the dog. Now, I know my body language. I'm a behavioralist. So you always take a step back and then come down a little bit, because you don't want to be over top of a new dog.
Right.
So come down, and I also kind of turn my body a little bit, because if the dog's going to bite me, I want him to bite me in an area that has got some good padding. I hate to say it like that, but I do. I want some padding. I don't want him to break my hand. I don't want him to get my face. And I got a lot of important stuff in here. So I turned slightly and hand under the chin, and the dog attacked me and got me and ripped and tried to eviscerate me. He did rip a big hole in my scrubs, had a big, huge bruise on my abdomen. If he'd gotten a better grip, he would have ripped out part of my belly. So I fell back and was like, okay, I'm gonna leave the room right now. Well, it turned out that this dog, this was not the first time he'd ever done it, and the owner never told us.
Wow.
The owner didn't tell us that this was a possibility. Same thing would happen with the Springer. And so this all comes back, see how I brought it back? Comes back to our genetics. The way that we line up these repeaters to have a dominant trait does lend the possibility for these little evil spirits to wiggle their way in.
Like Russian roulette.
It is literally like Russian roulette. You don't know. You might get the trait that you are looking for, but you have no idea what's come in the back door.
That's the funny thing about genetics.
Do you think that those 2 breeds have like some part of the same, since they're both in like the setter?
There's been so much research done on that.
Right.
Springer Rage, that's why it's called Springer Rage, because it usually only happens in Springers. The studies that they've been doing on it is now showing that it is a disease similar to epilepsy.
Okay.
And that the Springer Rage, the episode, the aggression, is similar to them having like a grand mal seizure. The same brain activity is happening when they go into a Springer Rage or a grand mal seizure with epilepsy.
Okay.
So there's so much about the brain we don't know, and now we're messing up with their DNA. And we just wreck them. We just wreck them.
Do you think it might be like because the Red Golden, the Springers have the liver red, I mean, just like
There's-
chocolate labs have epilepsy too, right?
Yeah, they do. Now they have epilepsy, but not the rage. Rottweilers are prone to the epilepsy. I mean, that's what gives a breed predisposition to diseases.
Yeah.
Those are the evil spirits. Labrador retrievers, I say lab, you say hip dysplasia.
Right.
You know, you have Yorkies. I say Yorkie, you say luxating patella. This just happens. Dachshund, IVDD. Did you learn your abbreviations, IVDD?
Yeah.
Intervertebral disc disease. So you've got these breeds that we have the evil spirit came in the back door when we got the trait that we wanted. So this is kind of, we just wreck them. We just wreck them. We take these beautiful dogs and we go like, let's change it, and then we wreck them.
There's like,
It's
there's
so
like activists
bad
saying.
that they not breeding. Like,
Oh,
they
hang
have,
on one
like,
sec. I got one that started up
This
here.
is where your animal activists come in saying you got people trying to play God, trying to make their own animals because we're always trying to do, we're mainly experimenting with them.
Pretty much, and because the canine is the only species that has this slippery property to their DNA that allows it to so easily be changed. There's no other
Well, that's
animal on the planet. I mean, you can breed a cow, and we got lots of different types of cows. We got Longhorns and Brangus and Angus and
But with it, you still gotta be careful, cautious because there's some like you got like the Chihuahuas and Pit Bulls. You can't terriers. You can't mix because their hands are too big while giving birth for the little Chihuahua.
oh my God, we have a whole ton of breeds that are 100% man-made. All of your bobble-headed dogs, you've got one of them on your mask right there.
Yeah.
Frenchie. Frenchies can't give birth naturally.
Right,
They can't even conceive now.
They can't conceive, neither can English bulldogs.
right.
They have to be IA. Yes.
I was just wondering, like, how you saying it messes them up or whatever, will they make it illegal if you, like, if they breed dogs that mess them up? No?
No. There's no way to regulate that.
Yeah.
There's no way.
I was just wondering if it makes a bad breed, well, I get it. Yeah.
But it's a crapshoot. I mean, somebody is saying that you got a red golden doodle who's, the picture perfect of health and stability. Knowing that evil spirit could be lurking in the background though. So you just never know.
Yeah.
You could have a red golden that never, ever, ever shows any aggression. But the idea is that they can't,
Yeah.
because it is that evil spirit that's attached to the red coloration.
So they're
Was that the
liable.
same thing with spider morphs and ball pythons?
What?
Is that the same thing in spider morphs and ball pythons when they get neurological disorders?
Yeah, like the
Oh, I have
bobbing
no
head, like
About any reptiles. We don't talk about no reptiles in this class, especially the ones that don't have legs. Honestly, the person to talk to about that would probably be Dr. Mendoza. He's our exotics veterinarian. I know that Ms. Regalia has exotics, but I think hers is wildlife mammals.
Yeah, Ms. Rivera has things.
Oh, yeah, no, we don't say the word. So, okay, so a little, here's, yeah, okay, now you're saying it again. Okay, no, in all seriousness, like 100% serious, and I'm even putting this on a recording. I am phobically phobic of that particular reptile to the point where if you continue to say the word, I will go into a full-blown panic attack. So, like it's a serious.
In all seriousness, chill the word.
It is a debilitating phobia. And I just gonna stop talking about it now. So that's just a little something about me. I'm not trying to be funny. I'm not trying to be rude. We can't say the S word because my heart's already, I'm splooshed out. So much cortisol right now. Oh my goodness gracious. So any of that, you can talk Ms. Rivera, Dr. Mendoza. If it's got legs, I'm fine. I don't maybe want them on me, but I'm okay. No legs, I am in a world of panic. Like urinate in my pants and fall down, probably pass out, rupture all the blood vessels in my eyes. Whew, I gotta sit down for a second.
I see the anxiety in you.
No, I don't.
You're looking flushed.
Oh. no joke.
I see that, I see that.
All right. So we have these nucleotides. We know that they bind together and that forms that double helix, that typical twist that we see. And that is our unique genetic code for every single individual. Now, some species, we do have commonalities. Obviously, we as humans have a lot of commonalities. Like our basic anatomical structure. We all have 2 eyes. We all have 2 ears. We function essentially the same. However, we all have very, very different genetic codes. So there are some things that will be the same across the board. Genetically, humans are programmed to have 2 eyes and 2 ears and 1 nose. Now, when the genetic stew gets stirred, that's when you start getting the mutations. You start getting what is literally termed, medically called a monster. A monster is anything that goes against the natural anatomical structure of the being. I used to have the dog that was the love of my life. You know those dogs? The love of your life, your canine soulmate. You never, ever, ever get over their death, ever, no longer, no matter how long it's been.
Yep.
Mine was a dog named Bentley. And he was a little mutt terrier, but he had 5 legs. He was a tripod. He had 3 fully formed legs, and he had 2 little chicken wings that came out of this one shoulder.
Oh my goodness.
One of them was kind of bent like this, and it was stuck. Well, it was actually just, he had no humerus. He had no radius ulna, so the whole part of his leg was missing.
Just a stump.
He had one little toe that came out of the shoulder, and that one he could kind of move it like this. And then he had another one that was just 2 toes, and that one was floppy. You could take it and turn it like 360 degrees. And so he was a mutant. He was a monster. He had 5 legs. Oh, he was a great dog. I lost him about 2 years ago. There's our DNA structure. RNA is how we get messages out of the nucleus. DNA is the king of the castle, lives in the castle of the nucleus. And the way that we get messages out to the body is our little messenger RNA. DNA is going to copy down what we need. He gives it to RNA. RNA then leaves the nucleus and takes the instructions wherever it is he needs to go to get it satisfied. So we have transfer RNA, which is what helps DNA copy down the instructions, right? We're transferring the instructions. Messenger RNA, just like it sounds, takes the instructions and runs them out of the nucleus. And we have ribosomal RNA, where we're delivering the instructions to the factory worker, and they can now create whatever it is that we need to create. So those are the 3 basic forms of RNA. That's it. And we know that thymine is replaced with uracil in the RNA. Give you guys a minute. Hey, did you guys do, is it your test today? Oh,
For internal and
Next,
external.
for internal and
We
external?
have it, it starts at 3 tonight, and is open till the next.
Oh, gotcha.
Even I know.
Huzzah! Huzzah! Skin. Nice. I'm trying to think of some good ones. Tongue. Both of you are correct. Lingual and glosso. Lips. Labio.
Isn't there another one?
So did I.
Or is that extra?
Kylie?
No, it's like something like.
Chilo.
Yeah.
Chilo kind of depends on who taught you. Eyelid, blepharon, tummy button, yeah, omphallo, omphallo.
Like umbilicus.
Yeah, sorry, your belly button. I call it a tummy button. Your claws,
Onkyo.
onkyo. How about testicles?
Belly.
Phallo's gonna be penis. Orchido. The reason for that is because, okay, remember I brought in the testicles for you to look at?
Yes.
And the way that they're formed, the way that they're kind of that oval egg shape, but they have the epididymis, they look like a non-bloomed orchid.
Yeah.
They look like an orchid that hasn't bloomed yet, and that's why orchido.
Oh, I heard.
That's how I was trying to connect it.
The flower,
The flower.
the flower of the testicles. How about uterus?
Sister.
Hystero or metro. Good job. Blood.
Hema.
Hema or hemato. Good job. All right, picture DNA. Here's just for you little visual learners. Here we've got the nucleus. Here's our DNA. Big queen bee right there. Holding the strings, holding the blueprints for everything. We're gonna get a message, hey, we need something. We're going to transcribe it, which means making a copy. We have this. Now our messenger RNA leaves the nucleus, finds a ribosome, and once it finds the ribosome, it is now classified as ribosomal RNA. These are just classifications. Same DNA. Here we've got our transcribed DNA, or RNA rather, transcription DNA, I keep saying D. Transcription RNA, messenger RNA, ribosomal RNA, and it's just telling you where is he and what's he doing. So then he feeds the ticker tape through, and the little ribosomes go, I'm gonna read this, do, and then it assembles the protein that we need based on the ingredients list that we've been provided. And this is happening in every single one of your cells.
So it makes
So
proteins
wow.
by doing that process?
Do what?
It makes proteins by doing that process?
Yeah, we can make tons of proteins, lots of different proteins. Because remember, proteins are the most abundant thing in your body. Everything from your actual DNA to things like hemoglobin. Hemoglobin is a protein in your red blood cells that binds with oxygen and carbon dioxide. Our hair, skin, and nails are made of protein. Epithelium. So this is just how our body says, hey, we need to make this thing, whatever it is. And the ribosomes are primarily just grabbing free amino acids and then assembling them per their instruction. Here's just another little cartoon picture. Here's our DNA, and we're just whipping up, we're kind of dividing, so we're like, okay, here's the part of the DNA that contains the recipe that we need right now. So we kind of unzip the DNA, and we make this copy. Then the copy leaves the nucleus, finds a ribosome, a little ribosome, binds and reads, do. It's like a ticker tape. Do. And you can see that now it's assembled whatever protein it's been told to. I know. Oh, you have no idea. Just wait. Anybody who's ever been in even the most basic biology class has heard the 3 letters ATP. Don't panic. I know this is another one, ATP, end up in the fetal position over in the corner. ATP stands for adenosine triphosphate. Basically what ATP is, it provides energy for the cells to do their job. What happens is, in a nutshell, we have these 3 phosphate molecules bonded together. One of them breaks off, and we now know what happens when we break a bond. We release what? Energy. So by breaking that bond and releasing a phosphate, we turn ATP into ADP, adenosine what? Diphosphate. Di meaning 2. And so now I've released a little blip of energy that gives the cell the energy it needs to do its job. And then we go ahead and pick up that 3rd phosphate, reform the bond, break the bond, release the energy. Reform the bond, break the bond, release the energy. That's how our cells get the energy that they need to do the work that they need to do. That's it. That's the whole premise behind ATP. That's it. We'll go into a lot more detail when we get into chapter 3 coming up here in just a minute. Because we're going to talk about the anatomy and the physiology of the cell. Yeah, we're going to be doing organelles, you guys. Don't you groan at me.
So the ATP is like gas for the car?
That would be an accurate way. It's giving energy so that it can do its job. I'll give you guys a minute. Furiously writing. What's the medical term for cell? Site. Nose. Rhino. Naso. Liver. Hepat. Remember, what are we gonna give ourselves this weekend? Woo! Little bit of hepatitis.
Yes.
No?
Yes.
No hepatitis for you this weekend? I'm gonna be giving myself full day. I'm tired.
She's right.
Yeah.
Miss Huff,
Yes.
if drinking gives you the hepatitis, what good does the red wine do when they say drink a glass is good for your heart?
Oh, okay, so everything in moderation. Mhm. Red wine contains something called an antioxidant because of the grapes. Antioxidant, every moment of every day your body is experiencing damage. There's a damage report being issued at all times. Right now, you have the sun beating on your foot, and that's damaging the cells. You have the fluorescent lights damaging the cells. You have simple radiation going outside, damaging your cells. Right now, all of you are occluding the small capillary vessels to your buttocks and the backs of your legs. Cellular death. You're killing cells right now. Killing them. Every time you're well, I guess technically anything that is on your body could be damaging cells. Whenever you get cellular damage, you get these little evil guys called free radicals. Because misery loves what? Company. Free radicals go charging through your body, just domino effect, making more and more damage. Antioxidants come in and bind with the free radicals and stop them from doing any more damage and allow your body to repair what damage they had done. So that's in the red wine. That's the antioxidants that are helping to repair the cellular damage that you've done just by living day to day.
But red wine is more potent than white.
Because of the type of grape that is made, like for example, there are foods that are very high in antioxidants. Blueberries,
Yes.
super high in antioxidants. Red grapes, super high in antioxidants. Those are the two that I can think of off the top of my head. So not everything has the same level, but it just so happens that the grapes associated with red wine happen to contain the high levels of these antioxidants. So now that we understand how bonds work and what happens when they're broken, we can now start looking at the associations with like different drugs. How do drugs react and work in your body? Different kinds of chemotherapy. How does that work? Different chemical reactions. And it's the breaking of these bonds that is also going to be very important when we get into nutrition. When you eat that Chipotle burrito and then poop out that Chipotle burrito, there is a whole lot of stuff that happens in between.
Burning going in, burning coming out.
Well, okay. you went there.
Yeah. Spicy.
But we're going to talk about digestion and breaking the bonds of our nutrients. We're going to be talking about how does that contribute to the growth and repair of your body. We're going to talk about like how does it contribute to your nerves sending messages to each other. And that's what gives us sensory to your brain or gives motion from your brain. So it's all about these bonds. It's all about these tiny little cellular bonds. And I think if I'm not mistaken, that is the last slide. We did it! We did it, guys! All right.
When we start chapter 3, there's 2 different parts to it, correct?
There is part 1 and part 2, and let me tell you why. So it's all chapter 3, right? Because we're going to be going over, what are we going over? Hang on. Phospholipid bilayer, the skin of the cell, and the organs. So essentially the anatomy of the cell. A lot of the pictures that I uploaded into the PowerPoint did not compress properly. So it made the PowerPoint like humongous, and it was too big to upload in one single PowerPoint. So I had to split it in half. That's why. It essentially part 1 and part 2, just the same PowerPoint. It was just the pictures were so not compressed that the file size was like, oh my God. And so I had to split it in half. So the place that I split it, part 1 is cell membrane, part 2 is organelles. So I've kind of given that natural division. And then chapter 4 is, so chapter 3 is what does the cell look like? How is it made up? Chapter 4 is how does it work? All right, so here's what I'm going to do now is I am going to cover that up and bring this over. And I'm going to call up, actually, you know what I can do? Just do this. There. So I'm going to call up some sample test questions. These may or may not be questions that you will actually see on the test. I don't know. I'm crazy like that. I'm crazy like that. All right, so this will give you an idea of the wording and how the depth will go. All right, shall I start with just an easy one? True or false?
False.
You got a 50-50 shot of guessing and getting it right. Anything that occupies space has mass.
True.
That is in fact true. Anything that occupies space has mass. I'll give you another easy one. Are you ready? True or false? In a strand of DNA, guanine will bind with thymine.
False.
Why is that false?
You don't put the tree in the tree.
Because you don't put the garage in the tree. See, those little mnemonics work. Sound stupid, but they actually really work. True or false? Temperature of the environment can influence chemical reaction rates.
True.
There, resounding true. All right. These are multiple choice, but I'm gonna ask them just as you tell me the answer questions, unless I need to tell you the choices. What element is a primary component of organic molecules? Carbon. Very nice, carbon. What structures are found in the nucleus of a cell? Protons and what? Nicely done, Zoomers. Protons and neutrons. And what is found outside the nucleus? Electrons. Which of these three structures contributes to the atomic weight?
Proton.
We got protons. Anything else? And our neutrons. Neutrons contribute to the weight. Why don't electrons contribute to the weight?
They don't have
Because
weight.
they're too small. So that was the atomic weight. Now let's talk about the atomic number. Oh my gosh, I didn't even get to ask it. I don't know what that accent was. I didn't even get to ask it. I'm just, don't even know.
He was even caught off guard.
Oh my goodness. The second electron shell will hold how many electrons?
8.
Nice. Eight. How about the first?
2.
Two. The pH of a healthy animal's body.
7.4.
Yeah, so just give me the average. 7.4. What does a buffer do?
It
does not
helps neutralizing acid.
Do what, Zoomer?
Helps to neutralize acid.
Huzzah! It helps to neutralize acid. When we talked about cows, and they have the buffer in their saliva, and they're constantly swallowing to neutralize the acid and prevent ulceration of their esophagus. Well, I used a lot of medical terms there.
So with the weak
That's pretty
acid, how
good.
does that?
Zoomer, what was that?
So with the like a weak acid, how does that neutralize a solution?
So anything that is less acidic than the solution it's being introduced to is going to neutralize to some degree the acid. It kind of depends on the concentration and the amount that you're adding. What is the term for decomposition of nutrients?
Catabolism?
Yeah, to break down catabolism. Remember cata, down under, and against, break down. Remembering the kangaroo, are we?
We didn't even see it.
And you didn't even see the picture. That's how powerful these pathways, these images are. What part of the atom has a positive charge? Protons. If an atom gains or loses an electron, what is it now called? An ion.
Oh, yeah.
Loses or gains an electron. It is called an ion. If an atom loses an electron, what happens to its charge? Yeah, so a negative, electrons are negative. Remember our bad feelings. What happens if I get rid of some bad feelings? I feel positive. If an atom loses an electron, how do I feel? I feel positive. If I grab onto that negative feeling and pull it towards me, now what has happened? I become negative. Sense. Think about those electrons like bad thoughts. here's an interesting one. If an atom has 5 protons, 5 neutrons, and 5 electrons, what would its atomic mass be?
10.
Yeah, 10. How did you get that? Protons and neutrons, add them together. The atomic mass or the atomic weight. Mass and weight, interchangeable. So if the protons and neutrons give you an atomic weight of 10, what would the atomic number be? 5. Good. How are atoms joined by a covalent bond?
Share.
Sharing is?
Sharing.
Sharing is? Sharing is? There you go. How do ionic bonds hold the atoms together? Yeah, electrostatic attraction. Just like pulling the sock off your pants. What are ions with a negative charge called? Those are anions.
Wait, no,
anion.
Very nice. Good job, Zoomers. How about a positive charge? Ions.
Cation.
This is the term that means a substance in which another substance is dissolved. Yeah, that's going to be solvent. And what do I call the thing that's being dissolved?
Solvent.
Solute. Nice. Guys, that's a pretty good representation of what you're going to see on the test tomorrow. That's about the depth to which we're going to go. If you study the testable materials PowerPoint, you're going to be just fine. You're going to be just fine. Where am I? Here we go. Chapter 3. All right. So now we've got a little bit of time left. I'm going to get started with chapter 3. None of this material that I'm going over right now will be on your test. This is all its own thing.
Is it cells?
If I can get this plugged in. Do I not have that the right way? I do have it the right way. There we
Is it
go.
cells?
Cells.
Anatomy of cell.
All right, let's start talking about cells. We've gotten our atoms, we've joined them together, we've gotten our molecules. Now we're getting into a little bit more of a complex structure. Cells. Cells are the basic units of all living things. You can have single cell organisms, they can live alone, or they can join together and make more and more complex organisms like us. We are put together by a massive, diverse population of cells. And every one of those populations does a different job. And that's the only job they will ever do for their whole entire life. A liver cell, now optimally, a liver cell should only ever do a liver cell job. A skin cell should only ever do a skin cell job. What happens if the cells decide not to do their job?
Cancer.
Bam! Cancer. Every cell contains a very unique genetic code. That's how your liver cell knows how to act like a liver cell. It has the instruction manual. Your kidney cell does not have the same instruction manual as a liver cell. Kidney cell has the kidney manual. Liver has the liver manual. And so when the liver cell then replicates, he passes that manual on to his children, or I guess technically her offspring. Although cells, I refer to them, I anthropomorphize everything. So I refer to cells sometimes as him and her. Cells do not have gender. And they're now little boy cells and little girl cells. Depending on their job, they can be very diverse in their appearance. A nervous cell looks very, very different from a nephron, which is the working cell of your kidney. So depending on the job, that'll define what does it look like in order to optimally do its job. We have multicellular organisms, for example, us. And this is where we have these little populations of cells that are all working together to form a task. And then ultimately their task hinges on everybody else's task to ensure that I am alive and well. If my kidneys stop working, that's going to affect every other organ in the body. So even though the kidneys don't directly have anything to do with the function of my liver, if the kidneys stop working, the ripple effect does affect all of the rest of my organ functions. So before we start getting into specific tissues, we need to understand the basic anatomy and functionality of the cell. So we're going to be discussing, let me go back for a second. Very simply, cells are built very similar to little people. Cells have a skeletal system, just like us. Cells have a GI system. Cells have a respiratory system. Cells have a brain. So if we start comparing the structure of the cell to the structure of us, makes it sometimes a little bit easier to understand how stuff works and why is it working that way.
Having a little
Now-
trouble reading the last line.
Oh, to understand, let me move that. So, like, am I saying that your cells contain gastric juices in a small intestine and they poo? Of course not. They do have a little void called a vacuole that functions just like our stomach. Cells do rid themselves of waste, just like us pooping. So essentially that's all we are, is walking sacks of cell poo. There you go. So, and that's all of us. That's not me calling anybody a bad thing. We are all just walking sacks of cell poo, because how many trillions of cells make up your body? Every one of them is pooping at any given time. Okay, but is it actual feces? Of course not. It's residual waste that is then eliminated from your body in one way, shape, or form. Ooh, man, we must have some sort of front or something coming in. Do we?
I think so.
We had
a mild one passed yesterday.
Oh, maybe that's.
That's
I don't know.
I have terrible. Oh, here's a good one. Osteoarthritis.
Inflammation.
Inflammation of what?
The bones.
Bone and
The joints.
yeah. So not only my joint, but also the bones get inflamed. And it feels, here's what it feels like. It feels like I have shards of broken glass
And they're all just
inside the
rubbing
joint.
together.
And the glasses are grinding inside my joint. And it's always a slight throb, but when there's a front coming in, oof, I can feel it. It's all residual from when I had the West Nile. We did have West Nile. And it disabled me for like 2.5 years.
Miss Huff,
Yes.
can you tell me how an ion is different from an atom?
Well, an ion is a form of an atom that has gained or lost an electron. So
Oh,
when
so
an
it's a-
atom, If an atom gains or loses an electron, we change its name to ion.
Okay, okay.
All right, so cells, let's talk about them. Let's get a little history lesson on the cell. The most basic, most primitive cell, our best guess, started about 3 billion years ago. But we don't know. Could be more. Could be 5 billion. And these primitive cells, they did not have a nucleus. That came later. However, these before nucleus cells, think of the nucleus kind of like the evolution of the brain. So when you have genetic material inside a nucleus that is functioning similar to a brain in a higher form. These little ancient primitive cells did not have a nucleus. They still had genetic material, right? They still had the blueprints for how to work, but it wasn't as organized. And they're called prokaryotes. Pro meaning before, karyote means nucleus. So prokaryote is before they had a nucleus. So they had this DNA, they had the blueprint, they had the instructions, but it wasn't contained in a separate area. And as this cell, this primitive cell evolved into a eukaryote, eu meaning good,
normal,
healthy.
good, normal, healthy. So we have now good, normal, healthy nucleus. Now our genetic material is contained, and the cell has evolved to something that is not nearly as primitive. Now here's a really interesting thing. Let me ask you a question. I'm willing to bet here's one that you already know. What do we consider the powerhouse of the cell?
Mitochondria.
Mitochondria. See, you all know. Powerhouse of the cell. Well, here's the funny thing about that. There's a lot of theories about this mitochondria. So mitochondria provides the energy for the cell, the organized eukaryote. But here's what we think happened. Because mitochondria contains its own DNA. Not in a nucleus, but it has its own set of instruction manual. It is the only component of the cell that has this. So here's what we think. We think that as the cell is evolving, changing, growing, prokaryote, okay, now I'm gonna act it out because it's kind of funny. Prokaryotes over here like, hey, how come you look so much more fancier than me? And the other cell's like, I'm evolving and I'm changing. And the prokaryote's like, well, I want to evolve and change too. And the other cell's kind of making fun of him a little bit. You know, well, you can't because you're just a primitive prokaryote. Suddenly this bigger cell, because the more organelles, the more complex, the bigger the cell had to be. So he's growing and growing, and suddenly this now complex cell is over here like, he doesn't have the energy because now he's super humongous compared to what he was. Prokaryotes over there like, hey buddy, what's wrong? Need some energy? I happen to have energy that I would be willing to share with you, but you gotta let me join your fancy complex cell club. And the cell over here is like, come on over, come on over. And prokaryotes is and joined up with the complex cell club. And that's what we think happened because when the cell needs more energy, the prokaryote can divide himself like an earthworm and create more of him to create more energy. So we think that that's how the mitochondria, now obviously, am I simplifying it to the nth degree? Of course I am. But we think that's how the mitochondria became incorporated into the complex cell, that it was in fact a remnant of the prokaryote. So it's a fun little visualization for you. It sticks. I'll give you guys a minute Furiously ready. If you means good, I'm gonna throw out a pretty provocative word. Euthanasia
Good.
means good death. Yeah, it means a good death. Good job. Foot.
Photo.
Photo. If I have inflammation of the skin of my foot, what?
Wasn't that literally on something?
Yeah.
Yeah, that was literally on our last quiz.
Photodermatitis. Stomach. Gastro. Intestines. Entero. The first intestine. The first part of the small intestine. It's the duodenum. And then. Ileum, I or E. It is actually the E, because remember, the E looks like a squiggle. Yeah, I is the bone, sassy bone. E, think of your intestines are all squirmy and wormy, and that's kind of the shape of a letter E.
Ew.
So here we have a picture, obviously not to size. Here we've got our prokaryote, and there's our DNA all squirmed up, but not in a nuclear envelope. Here's our complex cell. Everyone who has ever taken a biology class ever before in their life has seen this exact same picture. Right here, this little squirmy wormy like here, that is the size of this. This is the mitochondria. So now you can see the size difference. If this is the size of the prokaryote, and this is the size of the eukaryote, now you can see why the complex cell needed something to give them a little bit more energy. And that prokaryote, that mitochondria, contains his own instruction manual, contains his own DNA, and he can divide by fission like a worm and make more and more if the cell needs more energy.
That's the prokaryote.
Yeah, this is the prokaryote. Now in the cell, and this is just a theory, right? That whole scenario I just acted out for you. But we think that's how the mitochondria became the energy producing center of the cell. Was that through evolution, we've incorporated this energy producing being into the complex cell. So why are cells so small? Well, we do have some size limitations. Small cells are able to provide the nutrients needed. If you had a humongous single cell, if you had a single cell organism like the size of me, they would not have the ability to give themselves the nutrition needed to be alive. I am not a single cell being. I am a quadrillion cell being. And each of those cells is helping me to obtain the energy that I need to function. A bigger cell simply wouldn't be able to do that. They just can't take in nutrients fast enough, and the cell will die. Small cell is also able to be governed by one brain. Now, is that to say we don't have cells that have multiple nuclei? Of course not. We do have cells that have multiple nuclei. But smaller cells, the smaller the cell, the less nuclei I need. And so it's kind of the more nuclei, the more energy the cell needs, which means the more organelles the cell needs, and it just turns into a whole big thing. So that's why we have these size limitations. We're going to talk about the surface area of the cell and how do we get stuff in and out of the cell coming up here a little later on in this chapter. The cells have to be small so that they can effectively provide themselves with the nutrients and the energy needed to sustain. So our cell anatomy, we have the cell membrane. Sometimes we refer to it as the plasma membrane. The cell membrane is like your skin. It is what regulates stuff going in and out of the cell. The cytoplasm is all the crap on the inside. Cytoplasm is this jelly-like substance that the organelles float around in. It's everything that is making up. So if I am a cell, my skin is my plasma or cell membrane. Everything on the inside is now cytoplasm. And then, of course, my nucleus would be akin to my brain. So if we start comparing the cell structure to the structure of our cells, we have that now, that little stepping stone. That's what we're going to be doing. We're going to be comparing all of this stuff to things that it is similar to on ourselves. And time flies when we are having fun. Do I have any comments, questions, concerns? There we go. All right. So just so there is absolutely no test opens tomorrow at what time? 8:40, and it will close at 9:55. And it will close. So if you have not finished the
But we only got
test,
35 minutes on it.
you have 35 minutes.
God.
It is 50 questions, which is 45 seconds per question. It doesn't matter, because you're doing it on Moodle. So, and I know I've got like a minute left. So here's, I know some of you are like having a pucker moment when I say 45 seconds per question. I don't think you guys realize how long 45 seconds is. So here's what I'm going to do real quick before we finish.
Take your time, read the question.
All right, so here's what I'm going to do. I want everybody to imagine that you are working on one test question. It could be true or false. It could be multiple choice. One question. When I say now, you're going to start mentally reading the question. When I say stop, you will have finished one question. Are you ready? Start. You're still working on that one question. Still just working on that one question. Stop.
Well, if you're a slow reader, though.
45 seconds. But some questions are not going to take you as long to read as others. So normally, but pre-COVID, we give you 30 seconds per question. When you were taking tests on campus, we would time it out. So if you had a 30-question test, you'd have 15 minutes to do it. This is a 50-question test. Normally, I would have only given you 22 minutes to finish it. So I've given you over and above. So should be plenty of time. Test yourself questions, upload those to Moodle anytime before the end of class tomorrow. We will not be Zooming, and I will see you guys back on Monday. Well, I'll see you guys back on Monday.
Thank you.
All right. You guys have a wonderful, wonderful weekend.
You too.